Welcome back to Unit 3, Part 2 of the lecture presentation for SC090 Foundations of Science for Herzing University. As addressed in Part 1, upon completion of this unit, students should be able to identify the fundamental components of the cell, describe the function of organelles, integrate nomenclature or naming conventions of cell biology, and introduce processes of cell division. We're not going to do a real deep dive, that's saved for later courses, but you should have an introductory knowledge. In Part 2, we'll continue our study of cell biology. We'll take a deeper look at the organelles and how they help the cell get things done, how they get it right. We'll expand on the factory description provided in Part 1. We'll take a brief tour of cellular energy. We'll also introduce the stages and phases of cell division. These are big topics, and on our tour, we're going to keep it relatively brief. Remember that we'll be cruising over the surface of many of these topics, and you can further explore them in external resources like the Cellcraft game. So as we dive into organelles, we're going to start with the mitochondria. In the last segment, we introduced the big three. Now that we've built the cell, we need to ask it to do something. As will be a recurring theme in your studies, nothing happens without energy. Chemical reactions don't happen without energy either coming in or going out. Cells don't run on batteries, and we can't plug them into a wall. That means the cell must exploit chemistry to extract. Notice I didn't say create, because energy is not created, it's extracted from nutrients. This responsibility falls to an unassuming looking fellow called the mitochondria. The MC, as he's called, looks like a small brown bean lodged in the cytoplasm. This innocent looking guy is a mass of double-walled membranes which are folded in on themselves, and it serves as the power plant of the cell. The mitochondria holds specialized enzymes and chemical agents which systematically turn fuel, sugars, amino acids, and lipids, into energized molecules which can drive cellular processes. This is a very complicated and some would say boring process with multiple stages, steps, chemical agents, chemical helpers, and a whole lot of math. I had to learn it when I was in school and it was just way too complicated. I don't know that I've ever gone back to it. So we're going to just kind of blow right over it for now. We'll boil it down to this. The mitochondria takes a low energy molecule called ADP, adenosine diphosphate, and charges it up with energy to become the reactive molecule known as ATP, adenosine triphosphate. This molecule can carry energy to different cellular reactions, different locations, and then share that energy to drive chemical reactions before reverting back to ADP. Kind of like Popeye and spinach. So what we're doing in this process is transferring energy in the form of a chemical agent. So let's just draw it out real quickly. I'm just going to draw a big letter A for adenosine, and then adenosine is the chemical base here. And then we're going to pick green here for 1-phosphate, 2-phosphate. And remember, 2 equals di. So adenosine diphosphate is an adenosine with 2 phosphates. So then what we can do is add energy, in this case in the form of another phosphate, and we get, here's our big letter A. We'll go back to our green. 1, 2, 3. Again, this isn't the exact chemical structure. I'm just drawing for illustrative purposes. So 1, 2, 3. ATP tri equals 3. And this is a reversible reaction. ADP plus energy equals ATP. If you want, you can shed that phosphate, which takes energy with it, and it reverts back to ADP. So let's clear the board. Again, I want this to be clear. I'll switch back to black just to make it a little simpler. Oops. So I'm just going to simplify. ADP. This guy's reusable. It's kind of like a shopping cart. What we can do is pick up a phosphate, put it in the cart, take it somewhere else, and again, this is where it becomes ATP. ADP becomes ATP, and if we want, we can my primitive artistic skills, we can eject that phosphate, which again, energy, and the ADP goes back. Again, it's a carrier. It's like a shopping cart. ADP is a shopping cart. You put the energy in, you have the phosphate, you put the product in, the phosphate, you take it somewhere, leave it behind, the shopping cart goes back in the store. I hope this makes sense. We're not, creating or destroying. We're really just transforming. Empty shopping cart, put a phosphate in. Now it's energized. We can wheel it somewhere else and do something with it. In this case, check it out and take it home. We eject that energy. Shopping cart goes back in the store. The phosphate is the only thing that comes and goes. The shopping cart stays behind. All right, let's clean up our board and continue our discussion. Just like in a good lab, we always have to clean up when we're done. Oops. Let's bring that back. Sorry about that, folks. Okay. Next up, we have the ER, the endoplasmic reticulum. In this case, the ER isn't found in a hospital. The endoplasmic reticulum, which literally means twisted net of plasma, again, plasmic, plasma, reticulum is where we get the twisted net part, acts as a specialized manufacturing line within a cell. If you've ever watched a show like How It's Made, you can see how a factory line twists back and forth and kind of condenses itself for space. This is very similar. The ER has two divisions, much like a factory may have two different lines. Or if you're working with metal, you might have like a shaping line and then a sealing line or something like that. One side, the smooth endoplasmic reticulum, assembles fats, oils, and steroids. The other side, the rough endoplasmic reticulum, has specialized equipment, those little crinkled up balls of RNA called ribosomes, to help build proteins and membranes. Ribosomes, and remember I said I'd be getting back to them, look like small dark balls or dots within the cell. They're bundles of ribonucleic acid which help turn the instructions into meaningful products. They read the blueprint and then help organize the necessary parts. To help you remember, my wife loves to build Legos, but sometimes she struggles to read the instructions. So I act as her ribosome. I dig out the parts, I lay them out in an organized manner, and then help her create the actual building. We don't, have separate jobs. We work together. So we'll go back and make a picture of a cell, and I think we'll slowly start to fill in some of the components. So here we have the cell membrane. It's kind of curvy and looping, and we'll remember that this is that phospholipid bilayer. Okay? And then we've got in here the nucleus. We talked about him. Okay. Out here, we've got the cytoplasm. And now we've got our first addition. This is the endoplasmic reticulum. And remember, it's that twisted up thing like a factory line, and it runs back and forth. And part of it, anyway, has got ribosomes on it, those dots. And again, I'm trying to dot this fairly quickly for you, and I know you'll figure it out. So this is the ER, and this part is smooth, and this part is rough. And the way I remember it, R equals rough, R equals ribosome. Excuse me. And we also know that the rough also helps build proteins. Sorry, that one doesn't begin with R, but it'll help you come exam time, or at least I hope. So endoplasmic reticulum, big unified structure. Rough side, R is for rough. R is for rough, R is for ribosome, R is for protein. Smooth. Smooth is like slippery, slimy, right? So like fat, lipids. Okay? And we'll continue to fill in structures as we go along. Let's go back to the lecture. Next up, we have the Golgi apparatus. Again, this is kind of a tricky name, but it'll sink in eventually. After the factory makes the stuff, it needs to prepare the products for use and delivery. I'm sure a lot of you have probably worked maybe in a mail center or in a factory. I worked in a couple of different factories and stuff when I was coming up. And I know that the packaging is the responsibility of the shipping department. I think we all get packages from Amazon, and they're in boxes, and they're stuffed with the air things or the foam, and then they're taped and labeled across the top. That's the shipping department. In the cell, the shipping crew is known as the Golgi body, or the Golgi apparatus. Just like a proper shipping department, the Golgi apparatus knows what products are used internally and which ones are bundled up for export to another site. Some products are sent across the cell for use or conversion, while others are sent to the cellular membrane for release and transport. The Golgi apparatus is an educated shipper as it bundles different packages based on their final destination. See the slide for a quick review of the factory analogy. Again, quick summary. The foreman, or nucleus, foreperson, I guess I should say, holds the blueprints, DNA, sends a copy of the blueprints for processing. The manufacturing line, ER, builds new stuff, and the shipping department, Golgi apparatus, packs it for distribution. Some products stay in-house, the cytoplasm, while others are sent through security, the membrane, for use and export. So after the Golgi, after the ER, I'm going to draw a dotted arrow here. And I'm going to make this a squiggly guy. The squiggles help us remember that there's lots of tape and stuff involved. And this is Golgi. Golgi. And remember, this can eject products for export, or it can package things for use elsewhere. For example, if the nucleus needs materials for replication, it can go there. If the endoplasmic reticulum needs to repair itself or become larger as the cell grows, it can send materials there. It can even compile and assemble chunks of membrane to make the cell bigger. And remember, inside the cell membrane, we don't just have the phospholipid bilayer, but we've got these carrier proteins and these channels. We've got doors, gates, just like a factory. And these products can go out through these doors, through these gates, these channels, to the outside world. And it's packaged up and prepared by the shipping department. We're going to talk briefly about cell division. And it begins by realizing that nearly everything dies. Truer words have really never been spoken. Every cell has a defined life cycle, which it doesn't exceed a total organism. When the organism dies, the cells die. When you lose too many cells, you yourself die. So many cells need to be replaced due to old age. You've got cells that you start with and eventually wear out and are replaced. Think about your skin. Your skin is constantly turning itself over. In other cases, we need to replace cells that are lost or killed through injury. This is where cell division comes in. Cell division is defined as the process by which a cell copies its genetic material, divides its assets, and forms usually two new copies of itself. This process follows a number of clearly defined phases and stages. In a typical cell, this process is called mitosis and preserves the total genetic count of the cell. In specialized sex cells, this process adds a second division, which reduces the genetic material and produces 4 specialized cells through a process called meiosis. Why 2 cell cycles and reduced genetic material? If this didn't happen, the total genetic material would double with each generation. 23 pairs of chromosomes will become 46, then 92, then 184. Our cells would eventually fill up with DNA and wouldn't be functional at all. We'll move into mitosis next, and I'll try to provide some simpler drawings to carry us through it. Mitosis, an overview. The short version, because again, there's entire classes taught on this, of this process is still kind of lengthy. So the short version, still lengthy, but we're going to try to keep it manageable. We'll try to keep it bite-sized. Mitosis is divided into 5 phases, each ending with something phase. And then there's 2 stages. Stick with me as we'll take the 4-minute tour trip through mitosis. First, we have the longest phase, interphase. Inter literally means between time. This is the cell's normal functioning time. This is when the cell is just going about regular business. Interphase represents usually 90% or more of a cell's total lifespan, its total time. Remember that reproduction is just a limited function. So the normal stuff is just what goes on during interphase. It's every day. The first part of actual reproduction is prophase. It's important to remember that pro means for or moving forward. So this is the phase where division or actual reproduction really starts to move forward. We see the DNA condense into chromosomes, those little X-shaped guys that we see in cells. So we've got this increase in activity in the nucleus. The nucleolus disappears because the RNA needs to be parceled out and form new cells. The DNA needs to move around. And the nuclear membrane begins to disappear so we can split out the genetic material. Next, we have metaphase. I remember metaphase as met or meet in the middle. Metaphase, meet up in the middle. This bit of alliteration helps us remember that the chromosomes line up along the central plane. This is a lot like setting your money on the bank teller's stand so he or she can separate out the parts and give a full accounting. The genetic material is all present and accounted for, so it can split up in the new cellular halves. My key word for anaphase is away. A for ana, A for away. The genetic material and cellular assets move away from each other, traveling to the opposite poles. Remember that we're splitting the cell in half, and the different parts need to get to one side or the other so they're included when the thing splits down the middle. Much like splitting Halloween candy with your brother, everyone has their pile. Then we move towards telophase. Remember that you use a telephone to talk to someone far away. Telophase represents long-distance transfer of cellular elements. The parts begin to separate, pull apart, and the nuclear membrane begins to reform and collect the genetic material. It's kind of like reaching out and pulling your part of the Halloween candy pile over to you. The chromosomes start to get comfy and unwind. The nuclear membrane is reforming and collecting the genetic material. We're beginning to see organization with a new nucleus and nucleolus returning. Lastly, we split the actual cell. These two stages are called cytokinesis, or splitting of the cytoplasm. Cyto means cell, kinesis means movement, where it's moving into two separate blobs. And then cleavage, which big knife, big cutting knife, is a cleaver for physical separation of the membrane. Once this is over, we see two distinct cells representing new copies of the old cell. Each one holds a complete copy of the genetic material and enough hardware in terms of organelles to get started on cellular business. So let's just try to draw it out. And I want you to I want you to stick with me here, because you should be able to draw these guys out. So again, I'm just going to draw an eye for interphase. That's the cell just, kind of as normal. Normal business. P for prophase. The genetic material starts to condense. We're copying the genetic material because we're going to need to have two copies when we're done. Metaphase. The cell starts to elongate. Remember, M for meta. M is meet up in the middle. So we get one copy of the genetic material, two copies of the genetic material, and actually at this point we're starting to see chromosomes. So chromosomes are little X's of genetic material. They line up along the middle. We also see mitochondria, endoplasmic reticulum, Golgi, mitochondria, endoplasmic reticulum, Golgi. We're meeting up in the middle so we can divide things out. A, anaphase, away. Cell really starts to elongate. One set of chromosomes over here, one set of chromosomes over here. Again, the cellular hardware over here. Okay? And then we get into telophase. The cell's just about ready to split. We've got cytokinesis, which takes place, which is a blob of cytoplasm moves here. A blob of cytoplasm moves here. The genetic material condenses and we start to see a nucleus and nucleoli. Again, nucleus and nucleoli. And we've got an endoplasmic reticulum here and here. A mitochondria here, a mitochondria here. A Golgi apparatus, a Golgi apparatus. And then we've moved through cytokinesis and then we get to cleavage. And at the conclusion, the membrane seals off. And we have one cell, two cell, which are copies of the original. And at the end of telophase, we go back into interphase, which is, as we recall, normal business, the resting state. Does that make sense, folks? Again, I expect you're going to have questions. Make sure you connect with your instructors. Take a look over this. Look at the lecture notes. But again, interphase, prophase, metaphase, anaphase, telophase. We've got cytokinesis where the bits move apart, and then cleavage where it splits, and the process goes back to the beginning. Let's clean up our board and go back to our lecture. Okay, so we've talked about mitosis. I want to get into meiosis now. And again, I don't have a dedicated slide for this because we're going to do a lot of drawing. It's always been my preference to teach mitosis first and keep my explanation of meiosis simple. We just finished running through the phases and stages of normal cell division. This will help inform our position on meiosis. Here's the bare bones fact sheet. First, we get the first part of meiosis, which is basically a run through mitosis, a normal-ish cell division. We split the original cell into two parts, clean and simple, before things get a little weird. Upon completion of meiosis 1, the cell then doesn't shift into interphase. Instead, it gears up for another round of division. There's no time to duplicate DNA or create new organelles. The cell jumps right into prophase 2 and follows the same steps, except this time it only has one set of DNA to split up. This means the double-stranded chromosomes get split up and doled out to the offspring cells. Remember what I said about needing to do this to avoid doubling DNA each generation? This is where the cut down occurs. Because again, in reproduction, like in humans, a male cell and a female cell, a sperm and egg come together, and each carries half of the genetic code, and they come together to form one complete set of instructions for one complete being. At the end of meiosis 2, we have 4 specialized cells which carry a mixture of DNA from the individual's mom and dad, ready to combine with another specialized cell and form a whole new individual. So let's go back and run through the whole process, okay? So we're going to start mitosis 1. Normal cell interphase. Then we get to prophase 1, where we copy the genetic material, start to move forward, and get ready for replication. Then we move into IPM, metaphase 1. The genetic material condenses into chromosomes, and the organelles line up along the central plane because we're getting ready to split. Interphase, prophase, metaphase, anaphase. Again, the cell is starting to elongate, anaphase 1. So the chromosomes start to move apart. The organelles start to move apart. And we're going to get ready to split this thing. Then we get to telophase 1, where the genetic material starts to condense. The cytoplasm pulls apart, and eventually we end up with 2 cells. And I'm actually going to switch over to the eraser to represent that. So now we have 2 cells which are copies of the original. In the case of sexual cells, or meiosis, we go directly into meiosis or mitosis 2. So we don't go into interphase. No interphase. We go directly into a second prophase. But in this case, we haven't replicated the DNA, so we've just got chromosomes lining up along the middle. Those X's are 2 components, so they start to split apart. Now we've still got, some cellular hardware there as well. Okay? Then we move into anaphase 2. Again, the cell starts to elongate, but instead of having chromosomes, we have halves of chromosomes moving to each side, and then organelles. Then we move to a second telophase. Again, the genetic material starts to condense back into a nuclei, but again, each one is going to end up carrying a half load. We've got our organelles, we've got our cytoplasm, And eventually, now again, this is going to happen for each of these guys. So we're going to end up with 4 cells, each carrying a half load of DNA. And then these cells are going to be able to go on for reproduction. So this second cycle is where we cut down the DNA. We take the chromosomes, we split them in half, we dole out a half copy to each side, and then we're ready to pair with another cell for reproduction. Mitosis is pretty much always just mitosis. The difference with meiosis is there's no second interphase. Again, no second interphase, no replication. And we go directly into a second prophase. Remember, this is a second telophase. Okay, that's kind of complicated, but I think everybody can handle it. Review this, maybe watch this part a couple of times. Make sure you continue to use your secondary resources in the course. Stay in contact with your instructor, and keep fighting. We're getting near to the end. Thank you, everyone. We'll see you in Unit 4.